Hi, folks. Welcome back.
Hello.
Okay, welcome back, folks. I hope you had a nice weekend. It was kind of chilly this weekend, so I hope you're nice and cozy wherever you are. And just taking a second to get a few more students logged in. Okay. All right, we'll get started here. So welcome back. I hope you're doing well today. Looking at our syllabus, right? Looking at our syllabus, we are 2 weeks away from your second lecture exam. And of course, we basically, including today, we have 3 lectures because obviously the Thursday of Thanksgiving, none of us are going to be online as is appropriate. So we have 3 lectures to get you ready for your third lecture exam. Okay. I will release the study guide for lecture exam 2 this Thursday. I'll release it this Thursday, so that'll give you like a week and a half of preparation with the study guide, okay? Do you have any questions right now about our schedule, anything going on? Okay. All right, gang, so we're going to get into chapter 6 heavily today. And what I'm going to do is start with some of those calculations. So also, just to remind you, right, and I'll put this into the chat window, right? If you have your syllabus accessible, that's great. If not, I'll just tell you, right, on your syllabus today, I'm assigning workbook number 4. Workbook assignment number 4 is homework. Okay. Please complete workbook assignment number 4 for Thursday's class. The workbook I'm specifically talking about is the one that you had your unit conversion information in and the recent graph analyses that we did. Okay, so that's the workbook. Please do your best to answer assignment number 4 for Thursday. And we will go over it on Thursday as a group to make sure everybody's understanding. And so I'd like to begin with some extra practice on that because, you know, math isn't everybody's cup of tea, and I appreciate that that is true. So last time we were together, the very last piece of information I put into your notes were some formulas from chapter 6. Here they are once again, right? Formula number 1, final number of cells equals the starting number of cells multiplied 2 raised to the power of the number of generations. And we went through a little bit of conversation about what it meant to have a generation. Formula number 2, the number of generations equals, excuse me, I'm going to sneeze. Excuse me. The number of generations equals the log of the number of cells at the end of growth minus the log of the number of cells at the start of growth divided by this decimal 0.3010299, which I pointed out last time is actually the base 10 log of the number 2. And then formula number 3 to calculate generation time. Generation time is the amount of time it takes for a population of cells to go through one round of binary fission. And that equation is 60 minutes per hour multiplied by the number of hours of growth divided by the number of generations that have occurred. Okay, so before we continue, I have a question that I'd like to do some brainstorming on. And here's the question I'll put into your notes, and I would ask you to help me with some feedback through the chat window, or if you want to speak up, unmute your microphone and say something. Here's my question. Under what situations of life, under what scenarios, under what circumstances that we encounter might having knowledge of bacterial growth or really any microorganism be important? So why is it that these formulas, which I'm going to ask you to apply your mathematics skills to very soon, why is it important that we might want to know the final number of cells? Why is it important about the number of generations? What is important about the generation time? What aspects of our lives could knowing information about the growth characteristics of a microbe be important? And so I'll start with number one, and I'd like you to help me add to this list, okay? Knowing things about bacterial growth, or in general, let me put in general microbial growth, be important. One is if you want to manufacture a biological product. Example, Anheuser-Busch, right? Anheuser-Busch, the giant beer company, brewery. They really got to know how the yeast are growing when they're going to ferment the malt and everything that goes into making beer, right? So manufacturing a biological product like beer, super important to know something about microbial growth, okay? How about other things, folks? Under what circumstances of life? Okay, okay, great. I'm seeing some things in the chat window, and I'm going to document these things. Go ahead, folks, and look at your peers, because they're saying some really good stuff as I continue to mistype. Okay, all right. So we had here manufacturing a biological product like making beer. Chelsea says about food preservation. That's a good one, Chelsea. Kit, you talk about manufacturing a vaccine. Absolutely. Okay. So let me ask you this in the chat window. Once again, I can't type. My question is this: were bacteria involved in the production of our coronavirus vaccine? Were bacteria involved in that? Any thoughts? Here's my next question as you ponder that. Okay. Okay. Here we go. What type of molecule is inside the Pfizer and Moderna vaccine? What type of molecule is inside the Pfizer and Moderna vaccine? Do you remember from anything you've read or heard about? Okay, thanks, Kit. I'm going to run with that. It's messenger RNA. Messenger RNA. So guess what? To make messenger RNA, you need a DNA template, right? You know that. So I won't go into a long-winded spiel right now, but here's how they did it in a nutshell. Pfizer and Moderna scientists, they took the gene, the sequence of DNA for a specific viral protein. So the DNA goes to the RNA, goes to the protein. They took the gene and they engineered it into a plasmid. Plasmid. Where in our course have we identified plasmids? Where in our course have we identified plasmids? Okay, thanks, Taylor and Aubriana. Right, in the bacteria. Good job, Elena. Good job, in the bacteria. So the scientists at these companies took the gene for a viral protein and they cloned it. They engineered it into a plasmid. They took the plasmid and they inserted it into a bacteria. And then through processes I won't describe right now, they forced the bacteria to make lots of messenger RNA that was encoded from the template on the plasmid. So they turned the bacteria into a plasmid generating facility to create lots of viral messenger RNA. So they made the viral messenger RNA from the plasmid inside bacteria. And then they purified that RNA, put it in a little lipid bubble, which is not technically easy to do at any place. There's only a few places in the world that have that technology. And they assembled it together so that hopefully we could be safer and end this pandemic. Okay, so you're absolutely right. That was a long-winded conversation, but I want you to see the connectedness of the ideas you're being exposed to here in the course. So absolutely, Kit, making a vaccine is a beautiful example. I'm going back up into the chat window there, food preservation. Yeah, right, absolutely, Chelsea. I mean, right? Don't we want to have some idea of the shelf life of foods we have, right? How long can you, I don't know, how long can you keep a bread out of the refrigerator, right? How long can you keep a piece of bread out of the refrigerator? You know you're going to get mold on the bread if you keep it out of the refrigerator. Heck, if you put the bread in the refrigerator, eventually it's going to get moldy too, right? There are issues of food preservation. Canning, the canning industry has to be worried about, whether they're doing the canning process correctly, because if there's any kind of disruption in the canning process, the food might spoil. Well, how long is it going to take for the food to spoil? Depends on a lot of variables, such as the temperature of the environment, the particular species of microbe that got into the can of food, things like this. Absolutely. Okay. Let me come down here under manufacturing a biological product. So making beer, making a vaccine, I bet you some of you know someone in your life that takes insulin to manage diabetes. And today in the United States of America, all the insulin is made inside bacteria. It's all made inside bacteria and then purified in special processes so that it can be put into an injector pen so you can sell it to somebody and they can manage their diabetes, right? It's all made in bacteria. And the point of it is you're going to want to optimize the yield of the product, right? You're going to optimize how much insulin protein you get for the amount of time and energy and materials you put into allowing the bacteria to grow, right? Okay. All right, so okay, let's see here. Any other thoughts on things that intersect? In other words, I'm fishing for something. For example, did any I know that our most recent conversation with the necrobiome probably wasn't pleasant for some folks, but as we get past that incident, think about forensic science, right? Think about how long a decaying body has been there at a crime scene. Now, we can't get specific about, well, this person died at 4:57 a.m. on January 3rd. We can't do that, but we can give a reasonable time frame, which is helpful to investigators. Kate, you're absolutely right that the manufacturing of antibiotics is heavily involved with this. Chelsea, dealing with waste and septic systems, absolutely, right? The sewer system, things like that. Why are we having problems? Well, maybe we need to go figure out what the microbial population is in the sewer. So, public health from that perspective. Public health, sewer gas buildup, et cetera, et cetera. Okay. All right. Arlen, you mentioned figuring out how long mold has been in your home, right? Maybe you're doing a renovation project in your house and you discover mold. Now, honestly, most folks don't care how long it's been there. I mean, if it's there, it's there, and we don't want it to be there, right? Because we don't want to be breathing in mold spores and things like that. But obviously, there might be some desire to understand how long the mold has been present. You think about historical sites where you're dealing with historical buildings, knowing the age of the mold or the characteristics of the mold might inform you about the historical story surrounding a structure. Okay. All right. All right. So we've got a nice list here, folks. And the point I'm trying to make with your help and brainstorming, and you did a really nice job, is microbes are everywhere, right? That's been the topic of our course here this semester. Microbes are everywhere. Knowing something about them can be helpful. Okay. I would also mention here under public health, I would also mention food poisoning outbreaks, right? If you have a situation of a local food poisoning outbreak that seems to be centered around one of your local restaurants, if you identify what microorganisms are causing the issue, you might have an idea of the window of time that you can expect to see large groups of the population be getting sick, might allow the medical community to better organize their response to that situation. Okay. All right, so lots of things that can be done with some information about the growth characteristics of a microorganism. That's what you've shown to yourself through this brainstorming activity. So we have some equations here that I'm going to be asking us to apply. You're going to have to apply them in a meaningful way for workbook number 4 assignment as homework over the next 2 days, if you haven't already been working on it. And if you have, that's wonderful. So let's use the formulas. Let's practice using the growth formulas. Okay, and so I'm going to ask if you please, have your calculator out at your house, or okay, yes. Kit, I see your response. Kit, the growth formulas were sent out in last class notes, but I've also copied them again here in today's notes. So you'll be getting them again this afternoon, Kit. And if you're really motivated, they are also in Appendix B of your textbook. Okay. All right. So with that being stated, let's do a practice problem together in a friendly atmosphere, okay? And this is friendly. You're not taking a test. It's okay to be wrong. So what I'm going to do is give us a hypothetical scenario. And I'm going to ask you to apply some logical reasoning and a judicious use of the formulas to come to an answer, okay? Remember, when I give you a hypothetical scenario, which I will do on your upcoming exam, I'm not telling you that you have to use all 3 formulas to answer the question. Maybe 1 or 2 of the formulas aren't even relevant. And that's the point of the math and the logical reasoning. Do you know when to apply which specific formula? Also, if I present a scenario to you in a way in which you have found an alternative, logical way to arrive at the correct answer without even using any of the formulas I've provided, that's okay too. All I care is that you honestly get to the correct answer, okay? So let's do a practice question, and if you have your calculators available, that will be helpful. So here's a practice scenario that I'm going to define. Let's say that it's summertime and you and your family are at a cookout, okay? And so let's say accidentally, unbeknownst to you, you feed your 4-year-old daughter undercooked chicken, okay? And you feed your daughter this undercooked chicken at 5 p.m. in the evening. Early evening, right? And you, because we have, magical things we can do in hypothetical scenarios, let's say that you happen to know that the chicken that you fed to your daughter at 5 o'clock was contaminated with 1,000 cells of Salmonella typhimurium bacteria. Well, this is a common food poisoning bacterium found on chicken, okay? The chicken your daughter consumed was contaminated with 1,000 Salmonella typhimurium, which is a bacteria, cells. Okay. We know from the medical literature that this species of bacterium is capable of reproducing once every 25 minutes in human blood. Okay. All right, so you fed your 4-year-old daughter undercooked chicken, 5 o'clock at a cookout, a barbecue. Chicken contained 1,000 cells of bacteria. We know that the bacteria is capable of reproducing itself once every 25 minutes in the human bloodstream. Next piece of the puzzle. At 4 a.m. the next morning, your 4-year-old daughter stumbles over to your bedroom to wake you up. And before she can wake you up, she bends over and vomits on your head. Okay? I don't know about you, but children are not too good at making it to the toilet before vomiting. Okay? So I think we've all experienced that. So here's my question. And here's what I want you to puzzle out. I would like you to tell me how many bacteria are in your daughter's bloodstream, in your daughter's body at 4 a.m. Okay, that's what I would like from you. So before you all start rattling away, let me ask you a question in the chat window. Here we go. Here's the question. Would you like to do this by yourself or team up in breakout rooms? The only reason I'm suggesting breakout rooms is because it provides a way to actually help each other. Okay. So any other opinions on this? All right. It seems that you want to go it alone. I will respect that. Okay, folks. So here's what I'd like you to do. Right now, during class, I want you to try to solve this problem for yourself. When you think you've solved the problem, send me, not everybody, but send me in the chat window. Don't send to everybody. Send to me in the chat window your answer, and I'll respond letting you know whether you're right or not, okay? And we'll go on for this for 5-10 minutes, give you some time to wrestle with it, okay? So go ahead. Go ahead, do your best. Okay, so I've had several answers thrown out there. None of them were correct, so keep trying. Okay, keep trying, folks. I appreciate the effort, I really do, and I'm responding to you. Just give me a little time. I'm getting backed up here. Well, you guys are throwing out the numbers like a spreadsheet. Whew. Hang in there. I'm trying to get to y'all. Okay, folks, we're going to go for about 3 more minutes. And let me say this. So far, one person has gotten it right. One person has gotten it right so far. Folks, we now have 2 people with the correct answer. We have 2 people with the correct answer. 2 people with the correct answer. Ah, it's good stuff, isn't it? I can feel the cerebrum warming up. Okay, in about a minute here, we're going to stop. Okay, folks, hold on a sec. Okay. All right. So, folks, if you didn't get this right now, it's okay. That's why we're here together today, to try to move you forward, right? That's what we're doing. So let me tell you that three of you got the correct answer. All right. And so for the sake of everybody, let me just tell you the names of the folks that got it right, because maybe you would want to study with those people if they would like to study with you. Okay. So Taylor got the answer right. Chelsea got the answer right. And Afala Shadi got the answer right. So Afala Shadi, Chelsea, and Taylor got the correct answer. All right. So maybe at least one study session with them might be helpful to you. Okay. All right. So Chelsea, Afala Shadi, Taylor, would one of you be willing to speak to the process you went through to solve this? I'd like your fellow classmates to hear it from another student before they hear it from me.
I can if my microphone's working now.
Okay, Chelsea, speak nice and loud, but go ahead.
Sure. So we know we have a start of 1,000 cells, and we know one replicates every 25 minutes. So the time from onset, which is 5 o'clock p.m., till the time that we were given is 4 a.m., which is 11 hours. There's 660 minutes in 11 hours. So 660 divided by, hang on one second. 660 divided by 25 is 26.4. So that would be your number of generations. So you're going to follow the formula, go 1,000 cells times 2 up to 26.4, and that'll give you a very large number, which mine didn't put into scientific notation, but
Okay.
yeah.
Okay. So let me ask this in the chat window. Let me ask this in the chat window. Were you able to calculate the number of generations, which is 26.4? Okay, because that's really the critical point here. If you couldn't get that number, then that would explain why you weren't able to answer this. Okay, is there anybody that would like me to go over the logic of getting 26.4? Please speak up. Okay, all right, I'm gonna do that. Okay, and I appreciate it. I appreciate you being brave enough to say yes. I would like to see the logic of that. Okay, everybody deals with sort of math things differently, and there's no one right way. There are many ways to failure, and there are also multiple correct ways to success. So I don't care which correct path you take, as long as you can prove that this leads to the correct answer. Okay, so when I start a logic problem, when I start a problem like this, and I'm gonna draw this out here in the notes. When I start a problem like this, typically what I do is take the information that I've been provided and I restate it. In other words, what do I know? Okay, what do I know? And again, this is simply restating what the problem said. So here's what I know. I know that the generation time is 25 minutes, right? I know that because I set up here in panel C, we know that Salmonella typhimurium can reproduce once every 25 minutes. So that was stated in the problem. I know that. I know that. What else do I know? I know that the number of cells at the start of growth is equal to 1,000. How do I know that? Well, because in point B, it was stated, right? It was stated here that the chicken was contaminated with 1,000 cells. And what else do I know? Well, here's what else I know. I know through a little bit of deductive reasoning, right? My daughter ate the chicken at 5 p.m. I know that. And I know she vomited at 4 a.m. So 5 p.m. to 4 a.m. is 11 hours worth of growth, right? So that means the total time that this microbe has been growing in my daughter's body is 11 hours. That's 11 hours of growth. Okay, question coming in the chat window. Is everyone okay so far? Any problems that we should talk about with the information we know? Everybody okay? All right, all right. So then, you know, Chelsea basically said in words how she went about getting the number of generations, right? 26.4. You did a good job, Chelsea. I suspect that maybe it's just a little too fast for some people to soak up that process, right? So you have to ask yourself, what are we looking for? We're looking for the number of cells, right? What do I want to know? Okay. The cells, the number of cells, excuse me. Number of cells at 4 a.m. Basically the end of growth in our current problem. Okay, so that will come up. You ask yourself, okay, I want to know the number of cells. I want to know the number of cells. Well, which formula will give me the number of cells? Okay, well, here's a formula that'll give me the number of cells, formula 1, right? The number of cells, the final number of cells after a period of growth is equal to the starting number, which I know, don't I? That's 1,000 multiplied by 2 raised to the number of generations. But doggone it, I don't know the number of generations. Well, I have a formula for the number of generations, but to do that, I have to already know the number of cells at the end of growth, and I don't know this. Which means I cannot use this formula to figure out anything for this problem. I don't know that, so I can't use this. Well, okay, that doesn't help me. But what else do I know? I know the generation time, right? I know the generation time. I know the number of hours of grown. So if I just kind of work this logic out here, I should be able to get the number of generations that have occurred, right? All right, so we have 11 hours of growth, and we know that there are 60 minutes in 1 hour, right? So that means the total period of time of growth is 660 minutes. So this organism from 5 p.m. to 4 a.m. was growing for 660 minutes. Well, I need to know the number of generations, and I know that each generation took 25 minutes. So I'm going to take this 660 and I'm going to divide it by 25 minutes per generation. If you do that, 660, let's do this on the calculator on the screen. 660 divided by 25 is 26.4. That's how you get the number of generations. You've taken the total amount of time that the microbe is growing, put it in units of minutes, and then divide by the generation time, which was stated in the problem. And you get 26.4 generations. Now, I know all the information I need to solve Formula 1. So that's going to be equal to the starting number of cells, which is 1,000, times 2 raised to the number of generations, 26.4. So if you do this in the calculator, you're going to get the following response. And I'm rounding here a little bit just to keep it short. 8.86 times 10 to the 10th cells. That's equivalent to 88.6 billion cells. So the first point I want to make is in 11 hours, 1,000 cells of this pathogen have gone from 1,000 cells to just shy of 90 billion cells in your daughter's body. Now, obviously, the number here might be a little bit off because your daughter's immune system is going to try to fight this thing. So maybe this is not a perfectly accurate number. I see your comment, Elena, hold on. Maybe this is not a perfectly accurate number, but it is a reasonable number when you think about the logic of the problem. Maybe the number is actually 70 billion because your daughter's immune system has been at work. But your daughter's sick, she vomited. So that means she needs help, right? So, all right, so here's a question I want to help with a little bit. So to do this in the calculator, here's what I would do. Wish I had a different monitor here. Okay, so to do that calculation, here's what I'm going to do. I'm going to take 1,000. I'm going to multiply it by the number 2. And then I'm going to hit the power key, which in my calculator is x to the y. X to the y. And hit 26.4 there. And that should give me that big long number, which in this case, my calculator is spitting the whole darn thing out, right? 88.5 billion, okay? Hold on a second. I'm just responding to somebody in the chat window. Okay, so alternative buttons on calculators for power key. I see that's an issue for some of you. Okay, so here are the keys on a calculator, power keys. Each company of calculator manufacturer does it differently. So you might have X to the Y. You could have Y to the X. You might also have an upside down thing that's called a caret. Can you find any of those things? Can anybody think of another key that represents power on a calculator? Because that's what we need here. Okay. So I'm sure if you have a scientific calculator, you can do the power key. And if you can't find it, we're going to have to get together so you can find it. Okay. All right. So if my comments are speaking to you, if my comments are speaking to you in terms of trouble using your calculator, let's hang on for maybe 3 minutes after class today. Let's get this cleared up so that you can be successful. Okay. All right. So hang on after class, just a couple minutes. I promise to help you. Okay. All right. So here's our answer. So if you were to get that, good job. If you understand, even if you didn't get this answer, okay, hold on a second. Here it comes. And I don't want you to tell me yes just to make me feel good. I'm not interested in feeling good. I'm not interested in that. I want an honest answer. Okay. I'll follow Shadi. I see your comment. Just hold on a sec. Okay. Okay. Because I want to know what it is I need to do for you to help you. Okay. That's what I want. So we can talk more about this after class today if you need help. Now, for those that got the first answer correct, okay, so, this is something here which is a correct answer. And you don't have to worry about changing the number format. You can spit it out to me on an exam exactly how your calculator does. That's totally cool, okay? There is an alternative way to think about this to get an alternative correct answer. So let's do that.
I have a question quick.
Okay.
So the formula that we used, now will that be like, will we always use 1,000, or I'm sorry, the 2 times the, exponent of generations? Because like I got the generations just fine, but I wasn't sure if that was the formula that we should have went with. So I had just ended up multiplying 1,000 times the 26.4.
Okay, thanks for asking. Yes, that is where you made your error. That 2 is always going to have to be in that calculation.
Okay, because I when I got it wrong the first time, I was trying to figure out like how many times, or how am I trying to say this? Like how many cells are reproduced from the original 1,000 to come up with the correct final answer. But now I get it. We got to
Okay.
use the 2.
Yeah, and so if you look at the comments of your classmates in the chat window, they're actually very insightful. Notice that the number 2 is a constant in that formula. So you don't have the luxury of throwing that 2 out. That 2 is supposed to always be there if you're going to do this equation calculation. Okay. And no, honestly, the reason the number 2 is there is because if you think about it, every one cell, when it reproduces, it becomes 2. That's why that number 2 is a constant in this equation.
Got it. I was like literally trying to figure out how in my calculator to come up with like 1,000 plus 1, like thinking that it was going to add another cell every time it reproduced or something. So I get it. Thank you.
Okay, Rachel, no problem. So these are the kinds of things I want you to say, because, if we don't have honest conversations about the weak points in our logic, then we just don't move forward, right? We don't ever understand, and we continue to spin with that tar ball of anxiety about math, and we just don't want that. So an alternative correct answer here, which nobody came up with, and that's okay, because on an exam I will take one correct answer. That'll be good enough. Okay, but let's think about it. Okay, the number of generations that we've calculated above was 26.4. So here's a question coming in the chat window. Can you have 4/10 of a child? Will such a child survive and live? Okay. Now, I mean, that's a kind of a grotesque idea, right? Of course not. Of course not. That's not viable. So I'm here to tell you 26.4 generations, 0.4 of a generation will lead to an incompletely formed group of bacterial cells that themselves will never make it. They will never live. They will die. So what I'm saying is, if you think about it, 26.4 is the mathematically correct answer for the number of generations. It's mathematically correct, but it does not make any biological sense, because you cannot have living cells that only went through 4/10 of a generation. It's not going to work. Okay. So in such a case, 26.4, right? 0.4 makes mathematical sense, but not biological sense. So what I'm saying is that what I would do here is do the following. I would do the calculation where I round the generations down to the whole number, the integer of 26, and do it that way without the 0.4. Because again, cells that only go through 4/10 of a generation are not living cells. They will die. They're not viable. So if you repeat the calculation this way, you will get a similar but different answer from the first one, which is roughly 6.67 times 10 to the 10th cells. Okay, so you might ask yourself, well, which of those is really the correct answer? Well, they're both mathematically valid, but the one that is biologically valid is this one. And so you get to see that, math is the beginning of our wisdom. It's the beginning of our reasoning in science, but it's not the end point. Okay. So Yasmin, to answer your question in the chat window, if you have a scenario like this, you would be justified rounding down for the number of generations. Okay. I mean, even if it's 26.7, right? 7/10 of a cell doesn't make any sense either. So round down to a complete generation for your calculation. Okay, but honestly, if you give me either of these answers, if this was sort of the problem on an exam and you gave me either of these answers, I would give you full credit for it. Okay. All right. Any other thoughts or questions? Okay. All right, folks. My God, time flies when you're having fun, doesn't it? Okay, so let's continue here. But I wanted to do this to give you some foundation for your homework, and we're going to hit the homework on Thursday, and then I promise you I've got another practice problem after that. It's all coming your way. It's like a television show. It's coming, folks. Okay. Okay, so. All right, so that gives us a pretty good position to continue our conversation here. All right, so I'm going to get started with some lecture notes in chapter 6 here. Topic of chapter 6 is microbial growth. Now, some things are going to be pretty straightforward here. I think so. And so I would ask you to have your textbook open and available. Okay, so microbial growth. What must be true of the environment to support growth of a microbe? These are common sense things, I think so, such as physical requirements for growth. Number one, temperature, right? Temperature plays a huge role in whether a cell is going to be capable of surviving or not, or reproducing, right? Yeah, I see some comments in the chat window. You're absolutely right, okay? So temperature, okay? Now, when we talk about temperature in the field of microbiology, there's certain buzzwords, certain vocabulary words that come up and are juicy exam question kind of things, right? So take a look at figure 6.1. Take a look at figure 6.1. Okay, I'm not going to list all of these out for us, but as an example, we have different classification vocabulary. For temperature, which is to say temperatures that different types of microbes thrive in. Okay, so let me put a couple of these in your notes. Okay, and so just to move things along, I won't ask any individual person. I'll just try to get you all to participate here. Here we go. I'm horrible. I'm racing here. In figure 6.1, what is the optimum growth temperature for psychrophiles? Psychrophiles, what would you say? At which temperature do they have the most rapid rate of growth? Take a look at figure 6.1. What do you say there? You're going to have to estimate it off the graph, right? Okay. All right, thanks, Kit. All right, and everybody, thank you. Yes, I think 11 or 12 is probably what I'm fishing for there. You know, I'm going to go with 12, okay? So roughly 12 degrees is the optimal growth temperature for psychrophiles. Okay, so species of microbes that grow best in what we as humans think as cold temperatures, they're called psychrophiles, right? Remember, everything here we're talking about comes from the biased perspective of being a human being, right? I don't want to be outside at 12 degrees Celsius. That's cold. That's uncomfortable. I don't like it. So microbes that live in those cold temperatures, cold from the human experience, are given the name psychrophiles. Okay, so psychrophiles grow best at roughly 12 degrees C. We would consider these cold-loving microbes. So obviously, psychrophiles represent the category of microbes we need to worry about spoiling our food in our refrigerators, right? Because it's cold in the refrigerator, but these microbes actually like to live in that environment. All right, so if you get, I think you have the idea now. So I'll just put a couple other of these in your notes, right? Mesophiles. These are the microbes that like to grow best at approximately 37 degrees Celsius. And so you can appreciate that 37 degrees Celsius is roughly the internal body temperature of a human being. So the mesophiles are the category that we have to worry about in terms of pathogenic behavior. Okay, we also have thermophiles. I'm going to ask you, okay, so first off, we'll say that these are heat-loving microbes. Obviously, there are different amounts of heat, so it's kind of subjective and relative. But for the term thermophile is defined in your textbook, the optimum temperature seems to be around 62 degrees. Okay. Around 62 degrees. So let me ask you in the chat window, here we go. What process that you learned about in the virtual lab uses enzymes from thermophilic bacteria? Okay, so we've done a lot of lab activities this semester. There was only one virtual lab that you did where it was kind of like you stepped into a lab environment on the computer and you worked through things. It was the bacterial identification lab, right? Okay, good. Yes, Elena, Kit, you're absolutely right. It's PCR, the polymerase chain reaction, right? That process, which is the foundation of forensic analysis across the globe and all sorts of other things, that forensic analysis is only achievable by the process of PCR, and PCR can only be automated with robots if we have heat-stable enzymes, and heat-stable enzymes come from thermophilic microorganisms, okay? Provide heat-stable enzymes for PCR. Okay, all right, so I'm gonna go faster now, folks. I'm watching my clock here. So temperature obviously is a big deal. Think about it, you know, are you comfy and cozy in your house or outside in the wintertime, right? We know where we want to be. pH, another big consideration, changes in pH, that the environment changes in the environment that the cell is in, like the liquid it's in or the soil it's in. The change in the pH of that environment will change the shape of the molecules that are inside that bacterium or fungus or protozoa. And then those molecules change shape, they change function, and they don't work as well as they're supposed to work, right? So here's a question for you. What is the physiologically healthy pH of human blood? I know I've got some A&P gurus in here, so, right? Okay, so remember it's 7.35 to 7.45. There's a world of difference between 7 and 7.35 because remember pH is a base 10 logarithmic scale. Base 10 logarithmic scale. Okay, so changes in pH will alter the shape of molecules, which you know changes the function of molecules. All right. It turns out that many species of bacteria that we think about in science, which is not to say all species of bacteria on the planet, because that would be kind of vain of us to say so, but many of bacteria that we think about and deal with from a human perspective, most bacteria tend to grow at a roughly neutral pH range of 6.5 to 7.5. That is a fairly okay umbrella statement to make. So I'll do this. Many species of bacteria thrive in an environment a pH equal to 6.5 to 7.5. And just to give you some contrast, many species of yeast, which you remember yeast is a fungus, which is a eukaryote, they're more tolerant of acidity. So many species of molds and yeast, they prefer to grow in a pH range of 5 to 6. Okay. And so let me ask you this question, my A&P gurus. Is skin oil acidic, neutral, or basic? Okay, I'll run with the first answers, okay? You're absolutely right that it is acidic. It's mildly acidic, which is to say is what I'm trying to argue here is that, you know, when you get fungal infections of the scalp that give bad dandruff and things like that, it's because the fungus is living off your skin oil. It likes the acidity of your skin oil. When you get a case of athlete's foot, you know, because you've been working out at the gym and you've been walking into the gym shower room and all that kind of good stuff in there, it's because the fungus enjoys the acidity of the skin oil between your toes. Okay. All right. So another physical property to consider is osmotic pressure. Okay, pressure itself is resistance to change, and osmosis is the flow of water. So osmotic pressure is the pressure that builds up when a membrane tries to hold back water from flowing in a given direction. Okay. Okay, good to see you, Chelsea. All right. So osmotic pressure is the force, the resistance to the flow of water via osmosis. Okay, and so what I'm going to say is that most species of cells on our planet tend to like a roughly isotonic environment. Most cells on our planet prefer an isotonic environment. Okay. However, there are notable exceptions. Okay. And so let me ask this in the chat window. What was the name of salt-loving microbes that you had to know for lecture exam 1? Do you remember it? Okay, good. Halophiles, absolutely. Halophiles, right? Halophiles love salty environments. Okay, and so they have special changes in their physiology because of bacterium, special changes in their cell wall that allow them to survive in an environment where water of the cytoplasm wants to leave the cytoplasm and leak out of the cell. Most normal cells, like your cells and my cells, animal cells, you throw that kind of cell in a hypertonic environment and things are not going to go well. Okay. Okay, racing here. Next would be atmospheric pressure. Atmospheric pressure is the resistance to the motion of the gases that make up our atmosphere when they're moved by gravity, right? That's atmospheric pressure. At any rate, most of us live our lives on the surface of the planet, right? No, none of us are living a mile down underneath the soil. And for the most part, most of us don't live our lives up on the top of Mount Everest, although theoretically that would be possible, right? So most of us live at sea level atmospheric pressure, including the microbes on our bodies and the surface of the soil and things like that. Most life that we think about is at atmospheric sea level and pressure. Okay, but you can imagine, I mean, I won't ask you to contribute because we're out of time, but what's going to happen if you were somehow magically transported down 1,000 feet below the surface of water without any protective equipment, right? That's not going to be a good outcome for you, right? At the same rate, if you were to find a microorganism that naturally lived its life 1,000 feet below the surface of the ocean, and you suddenly brought it back up, it's not going to do well because it's going to go boom, right? So atmospheric pressure matters. And just to give you an example here, and then we'll stop, there is a treasure trove of life on our planet underneath the oceans. And there's a category of eukaryotic microbe called foraminifera. That have been discovered with research vessels living as far down as 7 miles underneath the surface of the ocean. And what that means is that down there, by the way, this is what these microbes look like. Here's just a quick snapshot of some of them. Okay. These microorganisms are surviving in an environment which has 1,100 times the amount of atmospheric pressure on the surface of our bodies, living, basically living at the surface of the ocean. They thrive at 1,100 times normal atmospheric sea level atmospheric pressure. Okay, so in those moments of our lives where we get to think about things from a purely intellectual perspective and not worried about getting sick from pathogens and stuff, it really is kind of amazing to think about the variety of life on our planet that live in very unusual environments that are toxic to the human experience. Life is amazing. Okay, it is amazing. All right, folks, I'm out of time, so I'm going to stop.